very best SNP, the most correlated SNP in that region, and then found out that there was one gene that sat near it, this gene called MIDF, and so we found the gene that's causing white coat color in these boxer dogs. And this is exactly the same process that I use in all of my research, where we're looking for genes that are causing something. I mean, white coat color we looked at, but we're looking at cancer, and we're looking at epilepsy, and we're looking at diabetes, and we're trying to find all of the different genes that are causing these diseases in dogs. So thank you very much for joining me today for this lesson, and I hope you've learned something about the genetics of dogs and also about what makes dogs special. Hi, thank you very much for your interest in my lesson on mapping genes in dogs. In this lesson, your students will get a chance to search through the entire genome of the dog and find a gene that's causing a trait, in this case white coat color. And in this lesson, they're going to see footage from the lab that shows robots and machines that we use to actually do this work, as well as do the actual testing for correlation to find the position in the genome that's associated with the trait, and then you go into the World Wide Web and using a genome browser to find the gene. It's probably helpful if your students know something about DNA before the lesson, the idea that it's made up of A's, C's, T's, and G's, and that differences in DNA in people sometimes cause diseases. And as far as supplies, you probably need, nothing much more than just paper, writing utensils, and the ability to print out or provide the handouts that go along with this lesson. And a board at the front of the room would also be helpful. So it's divided into 7 parts. The first part is just a very brief introduction where I introduce the idea of dog breeds and then give the students a short lesson talking about, which of these dogs is bred for which of these behaviors. It's just an introduction for the idea of it. And then in the second part, I start talking about where dog breeds come from, and it just ends with a handout that's, how do you make a dog breed example. And then in the third part, I discuss why the process of creating these dog breeds also leads to higher rates of diseases in these dog breeds, and why, and then finish that off with asking them to brainstorm about which diseases they think that dogs are going to be getting. And so then after they've brainstormed as a class, the fourth part starts out with the actual list of the diseases that are most common in dogs. And it turns out that these diseases are exactly the same diseases that people are getting, things like cancer and diabetes, which is why dogs are a really cool species to be looking in for disease genes. And then I get into, in part 4, it kind of ends just talking about why we're using dog breeds at all if they're the same as humans. Dog breeds, it turns out it's really easy to map genes in them because dogs in a breed are so similar to each other. So that ties back to the discussion at the beginning of the lesson about how breeds were created. And then I just talk very briefly about what the genome is. I avoid using the term genome. I just talk about the DNA that's in a cell and how much of it there is, and then end off that part asking the students to brainstorm about how much DNA they think there is in a cell, how many letters, and then how many differences they think there's going to be between dogs. And so then in part 5, this is where we kind of get into exactly what it is, how we're going to actually map this trait. And so I give the trait that we're going to map. I describe it. It's a white coat color trait. It shows some cute puppies. It's different between the solid colored puppies and the white puppies. And then I explain that in order to find the genes that we're interested in, we look at 170,000 different SNPs, or differences, positions in the genome that are different between dogs. We look at 170,000 of these positions and find the position that's most correlated to the gene, the position that's most correlated to the trait we're interested in. And then that part 5 ends off with a worksheet that just introduces the concept of correlations and gets the students to basically, it's a worksheet that says, what does it look like if you're 100% correlated? What does it look like if you're not correlated? And just to really make sure that they understand what a correlation is and what it is that we're looking for. So then in part 6, we go over this worksheet on correlation, and then we show how we're using this with the data that we've generated for these 170,000 SNPs. Oh, so I should mention that in part 5, that's also the part where we go over the lab work that's involved and have some footage of the robots that working in the lab and things like that. So a little bit of some shots of pipetting that we use to extract the DNA from the dog blood samples. But anyway, so in part 6, we're looking at actually mapping the locus. So we look at the data, we run it through the program that we use to find the association, we look at a graph that shows the correlation, and then we give them, finally we end off with a worksheet that basically at this point we've gotten it close to the gene. We haven't pinpointed the gene of interest, and this worksheet is what they're going to do to actually pinpoint the position in the genome that's where this gene is. And so this isn't actually a worksheet, this is a group exercise for the entire class where there's a big chart that's got 15 rows in it, and then the students break up into 15 groups, and each of those groups fills out one row in the chart. So you could draw the chart on the board, you could project the chart on the board if you had a projector, or on a whiteboard, and just get each group of students. They each have a worksheet, a handout, and each of their worksheets is slightly different from one another, so you do have to fill out all 15 rows of that table in order to get the exercise to work. Because then after they filled in the chart, and this is part 7, you'll find out that when the chart's completely filled in, there's only one position in the chart that's 100% correlated with a trait we're interested in. And at that point, we know where in the genome the gene we're interested in is. I show going to the genome browser, looking up the website that can tell us what's in the genome in that position. We look at that position in the genome, and there's only one gene called MID-F, and that's our white coat color gene, and that's where the exercise ends. And so basically it kind of starts with why dog breeds are cool, takes you all the way through the process of mapping a gene, which is exactly the same process that they use in human disease studies that you may read about in the news, where they're studying things like cancer and other diseases in humans, but we're just doing it in dogs. And then takes you all the way to finding an actual gene. And I should also mention here that the example we're dealing with all the way through here, white coat color, is a real example, and we are looking at real data. So this isn't anything that's been made up. This is a study that we published a few years ago. So it's a very concrete example of what we do. So anyway, thank you very much for your interest in my lesson, and I hope your students enjoy it. I think it's a lot of fun. Dogs are always fun to talk about, and it's a great example of some really cutting-edge science that's going on around the world today. So thank you very much.